Hey everyone, I'm Elad from Astrolabe Diagnostics, and I want to thank all of you for joining the webinar today. The original plan was to have an RTCA meeting at the NIH, but unfortunately, because of the COVID-19 situation, we had to change our plans a bit. So we're doing a webinar instead, and I want to thank Mary at UNC and Carl at the NIH for organizing this. And I truly hope that within a couple of months, things calm down and we can organize an in-person meeting. So I'm going to tell you about data analysis with Astrolabe, which is an online platform that we developed for the analysis of flow and mass cytometry data. And before I begin with Astrolabe itself, I want to discuss the motivation a bit. Oh, before I forget, since this is a webinar, if you have any questions, please use the Q&A button below. And you can just click there, type in at any time during the presentation. And at the end, I'm going to address the questions that came up. So let's talk about the requirements that come with a flow cytometry project. And I'm being vague here on purpose because the project has many costs affiliated with it. It could be time, it could be the kind of flow and bioinformatics and biostatistics expertise that you need, and of course it could be the money that you need to spend. And as the project becomes more complex, these requirements rise exponentially. And I think that most researchers are deeply familiar with this side of the curve, where you're dealing with low complexity cytometry, which I define as 12 markers or less, or you're dealing with a low sample count, let's say a couple of dozen patients. And for these kinds of studies, we have a pretty powerful suite of tools you could use. You could use traditional gating and other manual analyses, and the statistics involved are pretty straightforward. However, over the past few years, we're seeing the advent of more complicated studies. And these come in one or of two flavors. The first one is high complexity cytometry. And these are 15 markers or more studies that involve the CyTOF, the Cytocorora, the BD Symphony, and so on. And a second category are increasing projects with increasing amounts of samples. And for example, we're seeing many more clinical trials who are aiming to incorporate immune monitoring for the purpose of biomarker discovery or responder stratification. And these can have hundreds or even thousands of samples. And for these kinds of studies, traditional gating is simply unfeasible. It is very time consuming, and the operator variability is starting to have effects on the kind of results and analyses that you're going to see. Furthermore, there's many more statistical pitfalls that come with these kinds of studies. And a good example is multiple testing correction for high complexity cytometry. If you have a high number of cell subsets in your experiment, it is quite important to involve statistical multiple testing correction for your analyses. So there's this concept of an analysis pipeline, which actually comes from statistics. And the single cell data that we're getting from the flow cytometer or the mass cytometer is just a bunch of points, right? It's a huge table with numbers in it. An analysis pipeline is a structured series of steps, whether these are algorithms, tools, methods, protocols that help you take this data and convert it into meaningful biological insights, something you can use to impact patients' health. And in the context of flow and mass cytometry, the analysis pipeline usually has three major steps to it. The first one is cleaning the data. This is also referred to as pre-processing. The second one is identifying features. And in the context of mass and flow cytometry, features are often cell subsets or clusters. And finally, there's the actual statistical analysis. And underlying all these is a layer of visualization that helps you make sense of each one of the steps and the connections between them. And once you have this framework in mind, building your analysis pipeline is just playing the game of multiple choice. For each one of these rubrics, there's different tools and methods you could incorporate. For example, for cleaning, you could use forward scatter versus side scatter or live dead. Then for identifying features, you could use traditional gating or clustering. And for statistical analysis, for actually bringing the experiment design to your data, you could use t-tests or logistic regression or any of the more complicated models that, for example, come with the R programming language. Finally, for visualization, you could either use traditional figures, such as biaxial plots and heat maps, or you could use dimensionality reduction, and two popular methods for that are UMAP and TSNE. So I'm going to discuss an example pipeline that came from a paper that was published a few years ago. Just before I do, I see we have some new arrivals, so thank you for joining. I appreciate it, and I'm sorry that I couldn't make it in person because of the COVID-19 situation. Please note that you can ask questions by clicking on the Q&A button at the bottom. You can ask these questions at any time during the presentation, and I'll get to them at the end. So I'm going to discuss an example pipeline that came from a publication that was published a few years ago. This was part of the Human Immune Project Consortium, and in this study, 42 pediatric patients who were infected with the chikungunya virus showed up at the clinic in Nicaragua, and the blood samples were taken from them during two stages of the infection, during the acute time point and during the convalescent time point. The goal of this study is to compare the structure of the immune system between these two stages, and the researchers used a plethora of techniques, RNA sequencing, Luminex, and finally, mass cytometry. And for the purpose of this webinar, I'm going to focus on this last data set, which was acquired by Professor Adi Brachman in Mount Sinai. So the researchers had 84 samples, 42 patients times 2. How did they analyze them? What was the pipeline that they constructed? Well, for cleaning, they used standard CyTOF manual cleaning protocols. And I just copy-pasted a few lines from the methods section of this, of this paper. They normalized using beads. They identified cell events based on DNA signal and event length, and then they removed dead cells based on rhodium 103. So for anyone who've done CyTOF, these are pretty clear and pretty straightforward. It's really the standard for cleaning that kind of data. Then for feature generation, in other words, for identifying the different cell subsets, the researchers encountered a bit of a problem. And the reason is that, one, this was a pretty complex data set, 84 samples, each of which had 30-something markers in it. And also, due to the concern of batch effects, they couldn't just draw a template in Flojo and apply it to all the samples. So they incorporated many different methods. And briefly, they first used traditional gating with a few of the samples to identify major compartments. So all of the labels that you see in this figure, CD4 T cell, basophil, etc., these were identified using traditional gating in a small set of samples and then expanded to the entire data set using a classifier, which the researchers called node label. Then each one of these major compartments was clustered using a phenograph, which is a clustering algorithm for site of data. And finally, in order to align the clusters between the different samples, every sample was clustered separately, the researchers used an additional step of meta-clustering. And finally, in order to visualize all this, they used the dimensionality reduction algorithm called VISNI. And in this visualization, each one of the dots is one of the clusters that came from one of the samples. The colors are the meta-clusters, so the clusters that were aligned across multiple samples. And the labels, again, are the manually gated populations. So a pretty complex series of steps. And going back to the figure I presented at the beginning, since this is a complex project, it goes to this right side of the curve. which means that the time investment in identifying different feature was pretty substantial. The good news there is that once that step was done, the statistical analysis itself was pretty straightforward. In fact, all the researchers used was fold changes and t-tests. So in this figure, the x-axis is the different metaclusters that the researchers identified. The y-axis is the ratio in frequencies between the two time points. In other words, we're looking at the frequency or the abundance of each of these populations, and we're comparing that frequency between the two time points by dividing one by the other. And what this means is that all of these subsets that are in the red are higher in the acute time point compared to the convalescent time point. So for example, you can see that there's a cluster of classical monocytes and a cluster of intermediate monocytes, which are much more abundant in the acute time point. On the other hand, everything is blue is more abundant in the convalescent time point. And for example, you will notice that several T cells and B cells, notably this population of naive CD8 T cells, are all higher in the convalescent time point. So going back to the structure of analysis pipeline, here is how Michelmeier et al. Answered the multiple choice questionnaire. For the cleaning, they use standard site of cleaning. For feature generation, they use the combination of different steps and algorithms. For statistical analysis, they use fold changes and t-test. And then for visualizations, they use biaxial plots and the t-SNE dimensionality reduction algorithm. Now, full disclosure, I was actually involved with this project. So back in the day, about 3 years ago, I was working at Mount Sinai. And in order to analyze this project, we had a team of 2 people, myself and Professor Adi Brachman, who is an immunologist at Mount Sinai. We also had access to the Mount Sinai supercomputer, and it took us about 20 weeks of work just to get through this data. We had to spend about 12 weeks to import all of these FCS files and cluster them and run t-SNE. Then another 3 weeks meticulously annotating the different metaclusters, another 4 weeks for the statistical analysis, and then on top of this, we had a 2-week overhead of managing the FCS files, sharing them with different people in the project, discussing results with the PI, and so on and so forth. And I would say that this is not unusual. And again, going to the curve I've shown in the beginning, the more complex the project, the more complex the demands. And I think that people spending 20, 30, 40 weeks on their CyTOF or a data set is not unheard of. So let's switch gears for a bit and talk about Astrolabe, and let me explain what does our service does and how does it help you with your high complexity cytometry projects. Astrolabe is a standardized state-of-the-art analysis pipeline. So it follows all the steps I've shown you before, the cleaning, the feature generation, etc. But everything is done by the platform. It's done by different algorithms that we incorporated. And Astrolabe really works with your existing experimental workflow. So you get your specimen, whether they're human or mouse or non-human primates. You acquire them using your instrument of choice, which could be the Cytec, the CyTOF, the BD Symphony. Then the single cell data goes on Astrolabe, and the platform takes care of every step of the analysis pipeline, whether it's cleaning the data, whether it's identifying cell subsets and doing unsupervised clustering, all of the follow-up analysis. So Astrolabe also assumes the role of the biostatistician by doing differential abundance and differential expression analysis. And you will see that these 3 bubbles really correspond to the 3 steps I've shown you before, because there's no mystery here. There's no secret. There's no black box. We really are implementing the best algorithm, the best method for each one of these steps. The results are available through an intuitive interface. So everything is point and click. Nobody needs to know how to program. Nobody needs to know any bioinformatics. The analysis itself is fast. Astrolabe is cloud-based. Everything is on the Amazon cloud. So you can use the platform using your home laptop, and we will analyze your data in just a couple of hours. And finally, we're not some faceless software company. So if you have any questions about the analysis or about the results, you can always reach out to us. Questions like, which algorithms did you use? What does this analysis mean? Is this real signal or just noise? And so on and so forth. So I'm going to give you a brief demo of the Astrolabe platform itself. And once again, I'll remind you, if you have any questions at any time, just use the Q&A button at the bottom of your Zoom interface. So I'm going to give a demo of Astrolabe using the same dataset that I utilized in the case study. So this dataset is now publicly available. The team put it on Flow repository. And once again, this is the chikungunya dataset. And our goal is to compare these two time points, the acute and convalescent stages. What you're seeing here is everything you need to do in order to work with the platform. So first of all, you upload your FCS files. Again, Astrolabe is cloud-based. Everything is secure. Everything is confidential. Everything follow Amazon best practices. Then you set up your experiment design. And in this case, the experiment design is straightforward. We have 42 patients, and we have the two time points, acute and convalescent. However, this table is similar to an Excel spreadsheet. You can add columns, and you can add values. And no matter how complex your experiment design is, we can capture it in this table. Let's say you have different treatments. Let's say you have controls versus patients. Let's say you have 3 different stimulations or 4 different time points. All of these can be encoded here, and they will be incorporated into the statistical analysis. So you upload your FCS files, you set up the experiment design. It takes about 10 minutes of your time. You wait a couple of hours, and that's it. You can come back and start navigating through the results that Astrolabe offers. Our emphasis is really on getting you to these biologically meaningful insights. We want to help you publish the next paper, submit the next grant, and ideally reach the patients with your research. So I'm going to show you some of the ways in which Astrolabe helps you facilitate that. I'm going to switch to a tab called the Cell Subset Navigator. And this is a composite view of the entire experiment. Everything I'm showing you here is using all of the FCS files, whether you have 5 or 50 or 500. And the starting point for the Cell Subset Navigator is simply a list of all of the different cell subsets that Astrolabe identified in your experiment. We're using a combination of 2 algorithms. We're using the flow-sum clustering algorithm and the ACBalam labeling algorithm to automatically label all of the different cell subsets in your experiment. And the way we do this is using a series of rules that come from the literature. So once again, there's no secret here. These are pretty established rules, and the algorithm itself has been peer-reviewed and published. So everything is as transparent as we could have made it. And essentially, Astrolabe is going through a series of steps, which are very similar to traditional gating, only we're using clustering instead of gates. So first, we identify the CD3 positive cells, then the CD4 and the CD8, and so on and so forth. Next, Astrolabe takes this wealth of information and uses dimensionality reduction to present it in a more accessible manner. In this visualization called the MDS Map, each one of the bubbles is one of the cell subsets in the experiment. And the position of the bubble corresponds to the high dimensional distances between the cell subsets. In other words, if I color code this by CD3, you will see that all the T cells are next to each other. And then if I color code it by CD4, then here's the CD4 T cells. And then if you want to go somewhere else, I'll color code by CD20. Here is the different B cells. And finally, the myeloids and NK cells are up here. So really just a way to take all of the different cell subsets and put them on your computer screen in a way which is accessible and intuitive, so you can start interacting with them. Now, you can get additional information from this. For example, you can get the counts of these cell subsets in each one of the samples. However, for me, the exciting part is really the way you can overlay the experiment design on top of this visualization. So, as we discussed before, the purpose of this experiment is to compare the acute and convalescent time points. Astrolabe has a feature called Differential Abundance Analysis that lets you do that. So I'm going to turn on the Differential Abundance Analysis for the time point feature. And when I do, you will notice that the MDS map is going to be color-coded based on the fold change between the two conditions. In other words, the deeper the color, the greater the fold change between the acute and convalescent time points. So just by looking at this MDS map, you will notice that one of the subsets is bright red. And these are the same intermediate monocytes that we've seen as a conclusion from the paper that came from this study. I can click on them and scroll down to get more information about this subset. So first of all, to the left, I have a box plot that shows me aggregate statistics across all the samples for these subsets. Specifically, the white line is the median, and we can see how it starts at 6% for the acute time point and then drops by sixfold into 0.5% for the convalescent time point. Additionally, we can look at this data on a patient-by-patient basis. So over here to the right, I have a bar chart where every pair of bars is one of the patients. And we can see how overall each patient follows the trend that we see in the box plot. For almost all of them, there is a decrease in the frequency of the intermediate monocytes. However, we can quickly scan through this to find any patients that behave otherwise. And we will see that there's 3 patients. Here's one of them, here's the second one, and here's the third one, where we actually see an opposite trend. And there's an increase in the intermediate monocytes between the 2 different time points. And finally, everything here is quantified using a statistical test. So behind the scenes, Astrolabe is using logistic regression followed by multiple testing correction. And the FDR score that you see here is equivalent to the p-value. The higher it is, the more significant the results. You can take these figures, you can take these numbers, you can plug them in your next grant application because everything here is backed by the statistics. So Astrolabe is using the breadcrumbs that we left behind. It's using the panel to identify the different cell subsets. And it's using the experiment design to give you this kind of intelligence that really focuses on your research question. And as I've shown you before, these cells were featured in the paper that came from this study. Only back then took about 20 weeks of analysis. With Astrolabe, you can reproduce the results in 4 or 5 mouse clicks. So let me switch gears for a minute and talk about another benefit of using the platform. And to illustrate this point, I'm going to start from the naive CD8 T cells. And if I click on them and look at the different plots that Astrolabe offers, I will see that their frequency increases between the 2 time points. So it starts at 5% for the acute time point, then goes up to 10% for the convalescent time point. In addition to these summary statistics, Astrolabe offers a Heatmap that shows me the signature of these cells across the entire experiment. So in this Heatmap, every row is one of the samples, every column is one of the markers. One use of this is validation. For example, we can see that these cells are CD3 positive, CD8 positive, CCR7 positive, as we would expect from naive CD8 T cells. Another use is appreciating the heterogeneity in this population. So for example, if I highlight CXCR3, and I'm going to draw a little circle, a little square around it to make this clearer to see, you will notice that its value goes up and down. So different samples have different levels of CXCR3. And this hints that there might not be not just a change in frequency for this compartment. It's possible there's a phenotypic change as well, and CXCR3 is part of it. Now, in my mind, one of the exciting things about high complexity cytometry, once you go into higher dimensions, is the ability to include these additional markers that have biological significance, but are not as straightforward to understand, like CD14 or CD20, which has been characterized pretty well. So in order to incorporate these into the analysis, Astrolabe is offering a mechanism called cell subset levels. The bubbles that you see here all correspond to the assignment level. And these are canonical subsets. They come from essentially a literature review. The second level is called profiling. And when I switch to the profiling level, you will see that I have many more bubbles in the MDS map. I have many more subsets in the list up here. And the reason is Astrolabe takes each one of the cell subsets and further stratifies it using unsupervised clustering. So, for example, we still have classical monocytes here, but the platform has broken them down into 7 different clusters. Similarly, the bubbles here, they're still naive CD8 T cells, but we have 4 different clusters of naive CD8 T cells. They're also checked off in the list up here. Astrolabe offers a visualization called a volcano plot that lets you quickly assess the significance and the trends across a large number of clusters. So over here to the left, every one of the dots is one of the clusters. The y-axis is statistical significance. So the higher a dot, the more significant the results. And the 4 clusters that come from the naive CD8 T cells are already highlighted over here, and then 3 more dots down here. And you can see how one of these dots is much higher than the rest. In fact, it's one of the most significant clusters in the data. If we focus just on that cluster and scroll down, we will see a 10-fold change in frequency between the 2 time points. Furthermore, this trend is quite consistent across all of the patients. So just to recap, we looked at the naive CD8 T cells and saw a 2-fold change in frequency. But then, when looking at this specific cluster, we actually saw a 10-fold change in frequency, a much more significant trend. And this result was not reported in the paper that came from this study. In fact, I reached out to Professor Adi Brahman, who was in charge of the CyTOF in this study, asked him about the CXCR3 population, and he had no idea what I'm talking about. He actually went back into Cytobank and used traditional gating to indeed validate and verify that there is a CXCR3 positive population, which is almost exclusive to the convalescent time point. And I think this story illustrates nicely the benefit of Astrolabe when dealing with these large studies, whether you have a large number of patients or a complex panel because of the CyTOF or the CyTOF Aurora. It is almost impossible to cover all of the different trends and signals and hypotheses that come from the data. Even if you use bioinformatics, even if you use a variety of algorithms, there's already so much you can survey as a human. There's only so many hours in a day. Astrolabe is an unbiased approach to analyzing your data. The platform is really going to test all of the different clusters, all of the different subsets, all of the different hypotheses. And you can go to the platform and use this very friendly, very accessible interface to navigate through the different results. For example, you can say, I want to look at the top 6 cell subsets. And it's just a matter of clicking on them and then scrolling down the page to see what's the different trends, what's the signal in your data. All of this is very friendly, very quick, very accessible. So I'm going to provide a brief summary, and once again, a reminder, you can use the Q&A button to ask any questions. And I just want to go back to this slide that discussed the amount of time that we had to spend on analyzing this data set for this study 3 years ago. And I mentioned how back in the day, it took us about 20 weeks to analyze the data. Well, how long did this take with Astrolabe? So with Astrolabe, setting up the experiment is just going to take a couple of hours. A bench scientist using her laptop computer can upload the data, spend 10 minutes setting up the experiment, go have lunch, and by the time she's back, the analysis is done. And then she can share these results with everyone else in the group and navigate through all the different visualizations and analyses that Astrolabe offers to really try to find the most relevant hypotheses in the experiment and really decide what's the next step for her project. So once again, if you have any questions, please use the Q&A button below. And I'm going to take a couple of minutes to go over everyone's questions, and then we can answer them. Okay, let's go over a few of the questions that came up. The first one is, could you please give us a list of the algorithm used by Astrolabe? So it's actually a pretty thorough list, and thankfully the whole thing is available on the Astrolabe FAQ. So you can always go here and see what's the latest methods that the platform is using. And specifically for immunophenotyping, we're using a combination of two algorithms. We're using the flow sum clustering algorithm and the Ekbalam labeling algorithm. All of this was assessed and compared to manual gating in this paper that you see right here. For differential abundance analysis, we're using the EDGE-R package, and we're following the same methodology from Lunet L and from the DiffCite paper. So those of you who use the CyTOF might be familiar with these publications. And finally, the default cell subset definitions come from the Human Immune Project Consortium, specifically these two papers by Mecker et al. And Finnick et al. So the next question is, can you vary the number of clusters for unsupervised clustering and look at it dynamically, flip through the different versions? So that's a great question. And I think that when researchers deal with clustering in the context of flow and mass cytometry, there's always the question of how many clusters do we need? And in the case of Astrolabe, we're using the flow sum clustering algorithm. And there's a nice chunk of evidence out there that says that the best way to use flow sum is to over cluster your data. In other words, you want to have more clusters than you expect. And then you can go over and annotate them. And that's going to help you increase the chance that rare populations are going to have their own cluster. So what Astrolabe does is indeed over cluster the data. So for example, using the same naive CD8 T cells that I've shown before, it's quite likely that Astrolabe is using 10 or 20 or maybe even 30 clusters. However, for you as a researcher, navigating through so many clusters is extremely time-consuming. And one of the reasons analyzing this experiment manually took so long is because, if I recall correctly, we ended up generating more than 100 clusters that had to be manually curated. So what Astrolabe does is using a statistical heuristic to collapse these clusters. And what this heuristic asks is, do we need all these clusters? Are they meaningfully different with regards to the markers in the panel? So essentially, for every pair of clusters, if they look about the same with regards to the markers used, then they're merged. And you can see that for the naive CD8 T cells, we started with 10 or 20 clusters, and we ended up with 4, which is much easier for you to navigate through. And furthermore, you know that there is a statistical heuristic that was used to collapse them. Just to give another example, real quick, looking at the basophils, these were not clustered at all. And this is not because there is no heterogeneity in the basophil compartment. It's just that this panel doesn't have any basophil-specific markers in it. So we could go to FlowSum or Phenograph and ask them to cluster these cells, but it's going to be an empty exercise. There is no heterogeneity with regards to this panel in the basophil compartment, and Astrolabe helps you identify that really quickly. So to answer the question, you cannot change the number of clusters. The number of clusters is decided by the statistical heuristic. And again, the goal is to really guide you towards a best practice and trying to get the most out of your data as efficiently as possible. So the next question is, can we run Astrolabe with organisms other than human? And actually, this is a good a good time to point out the different types of samples that have already been analyzed with the platform. Astrolabe has been around for almost 2 years now. We've been licensed by many different organizations. And while this study is about human PBMCs, the platform has been used with human samples, mouse samples, and non-human primates. And furthermore, we've used it with PBMCs, bone marrow, whole blood, central nervous system, gut, spleen, lymph node, liver, and I'm sure I forgot 2 or 3 other tissues. And of course, in the context of both healthy individuals, infectious disease, autoimmune disease, cancer studies, immunotherapy, we've run over 1,000 experiments with Astrolabe at this point, and we've used them with a plethora of different model organisms, systems, diseases, and so on. Next question is, are there plans to make, if this is even possible, a version for single-cell RNA-seq data? So at the moment, Astrolabe is designed for panel-based data sets only. And we are using the platform for mass cytometry, flow cytometry, and we've also applied it to the panel-based element of CytSeq using the TotalSeq kit from BioLegend. We are looking into the option of developing a version for single-cell RNA-seq studies. We have the data science set up more or less, but we're still assessing whether there's going to be customers. So if you are interested in an automated tool for the analysis of single-cell RNA-seq data, please reach out to me because we're doing the market research now, and I will really appreciate any ideas or feedback that you have in this context. So these are the questions we have so far. So just another 10, 20 seconds. If anyone has any last questions, please use the Q&A button below. And finally, once again, thank you all for attending. Thank you, Marie and Carl, for organizing this. And I truly hope that in a few weeks, when things calm down, we can have an in-person seminar at the RTCA. So if you have any questions, please email me at this email address. You can also ask Marie and Carl to connect with us. And have a wonderful day and stay healthy and safe. Thank you, everyone.